All right, it's my pleasure to introduce our first speaker today, Clay Rains. He is a fish biologist at the USGS Eastern Ecological Science Center in Leetown, West Virginia, and a PhD candidate at West Virginia University. He previously worked as a fish biologist with the West Virginia DNR, as well as the Tennessee Aquarium Conservation Institute, where he oversaw restoration and monitoring efforts for threatened and endangered freshwater species, as well as conducted research on a variety of fisheries management topics. Clay obtained a master's in fisheries resources from Mississippi State University and a bachelor's degree in biology from Shepherd University, and he intends to defend his doctoral dissertation here next month. So good luck with that, Clay, and looking forward to your talk.
Well, thank you for the introduction, Nick. I unfortunately have a ton of time, a ton of information here and not a ton of time to deliver it. So I am going to fly, so I do apologize in advance for that. But thanks everybody for tuning in. I'm going to talk about a really beautiful gene expression data set, specific suckers with varying hepatitis virus infection states. I want to take some time to acknowledge my co-authors. Without them, none of this would be even possible. So let's get after it. Little background on our critter of concern, the white sucker, Catastomus commersoni. It's a benthic freshwater fish, pretty much ubiquitous in a lot of North America. It's got a good application as a sentinel species due to a benthic life history. So it's in constant contact with potentially contaminated benthos, and so a good candidate for studies of environmental contamination. Specifically, tumors or other deformities are categorized as beneficial use impairments, which in areas of concern kind of influence the listing or delisting of those particular locations. Major issue, particularly within the Great Lakes region. And this is not just a historical thing, this is ongoing. These pictures are from last week from a US EPA colleague who's actively conducting external and internal pathology checks of white suckers, probably as we speak. I'm not sure when they went out last. So this is historic and ongoing with a bunch of different implications. As a little bit more background, this is kind of a broader context part of the Great Lakes Restoration Initiative. So suckers are collected from all of these Great Lakes areas of concern listed there on the map. Specifically, I'm focusing on the Sheboygan River, where this data set was mostly sourced. And in addition to just general fish health, we also evaluated host response and then kind of a broad sweeping pathogenomic workflow to see if there were any new viruses that might contribute to some of these external and internal lesions and pathologies. And we were ultimately successful. Several novel viruses have been uncovered. Some of them have been published in literature, some of which have not, and all of which we're still investigating the significance. Relevant to today, we're going to stick with white sucker hepatitis B and white sucker hepatitis C, hepadnavirus and hepasiovirus respectively. And I will probably use those terms and maybe others interchangeably as we progress. First started with the hepadnavirus, hepatitis B, characterized by a bunch of folks on this call, along with Cassidy Shaw. Made great progress characterizing and identifying the first hepatitis B not from a mammalian or avian host. Subsequent efforts kind of broadened that phylogeographic understanding of the genetic diversity in general. Both of them also identified novel detection methods using quantitative PCR that influenced a bunch of different sample matrices and allowed for reliable and sensitive detection across a variety of different approaches. That was the first. It wasn't the last. We've continued to stack up hepadnaviruses in fish. A big burst came through in 2017 after the original in 2015. We put another one on the board just last year, and then another paper came out just yesterday, adding a bunch of other hepatitis B viruses within fish. So check that one out in scientific reports. And as DJ Khaled would say, another one. We also have evidence of a white sucker hepatitis C virus. This one's kind of an interesting story. It was sourced from that original Great Lakes data set focusing on the hepatitis B virus. And it was identified by Dr. Stefan Seitz and his team with the German Cancer Research Center. He developed a whole genome and identified a pretty good annotation, which we've been relying on. Subsequently, an Australian team published the exact same virus from the same short read archive reads, and credit where credit is due. Both of these are valid and relevant, but one is now represented in the literature, and we will be mostly relying on the annotation of Stefan Seitz. But it is out there in the public sphere now. So that's positive because this means I'm not breaking news in a student seminar. But the point is, who cares? This is kind of a little bit of a logical leap because we're still trying to puzzle this out ourselves. But the reality is that these mammalian orthohepatinoviruses are associated with hepatocellular carcinoma. That same connection in fish is still unclear, and that is why this effort took place in the first part. The lone data point that was originally uncovered did have a really high virus copy number of hepatitis B. However, we're talking slices of slices here. This is percentages of percentages. So in general, prevalence of white sucker hepatitis B across a couple of efforts was a little less than 10%. Liver tumors, less than 5%. Combination of virus and liver tumors, 2.4%. And then you add in prevalence or co-infection of hepatitis C, very thin slices of slices. And we're talking enormous sample sizes in huge amounts of time before you probably would even get any tumor formation, at least in mammals. So we're kind of a little bit hamstrung by logistical reality. But to try to answer these questions, we implemented a pretty multi-factored approach. So this particular data set, we collected 200 white suckers from the Sheboygan River. They were characterized based on their biology and internal and external pathology. Liver was collected. DNA and RNA were tandem extracted and subjected to qPCR or RT-qPCR for DNA and RNA viruses, respectively. From there, we were able to characterize viral infection as positive or negative for either Hep B or Hep C. Liver RNA, total RNA, was also subjected to nanostring code set gene expression analysis using an encounter sprint. And then we have this beautiful multivariate data analysis that we can use to build an association or resemblance matrix. We can do hypothesis testing, cluster analysis, ordination, all those fun things. More specifically, the kind of multivariate analysis we're using relies on what's called Permanova. Permanova is really an old principle built on Huygens theorem, which was actually described prior to the description of gravity, theory of gravity. And essentially what it is that you can build these multivariate data sets, layer them on top of one each other, and then through permutation correcting, you can build a data set that can be mapped in three-dimensional space based on similarity or dissimilarity between points within the same data set. So using that model, we summarized all of these data and found by doing this, you're able to generate a test statistic and a p-value for statistical significance. So in doing this, we were able to identify, unsurprisingly, that biology plays a big role in white suckers. Age, length, weight, and sex biological parameters were significantly different in this data set. This makes sense. They are sexually dimorphic animals, with females typically larger and heavier across same age classes, and all those things are correlated. Interestingly, liver pathology was not statistically significant, but a few external characteristics, white lesions and papillomatous lesions, were identified as statistically significant. Significant and biologically relevant are different things, but we were hoping to get that at least as a start. Taking a quick peek at this external pathology data set. In general, most of the pathology came from fish that were virus negative, did not detect presence of either virus. However, there was some external pathology indicated in fish with the hepatitis B and Hep C data sets, not so much in the co-infection data. We used a series of different mRNA probes with the nanostring, including 6 that were virus-specific targets. And thankfully, all of them were significant and relevant to our data set and able to be applied kind of as a suite of characters. Similarly, we had 41 biologically relevant genes that we targeted via these probes. If they are denoted here in bold and italics, then they were also significant. Significant and relevant, once again, different. However, we're relying on significance for this first preliminary understanding. And it was so many that I couldn't fit it on one slide, so there's a pile here. The only thing I wanted to really call attention to is vitellogenin, for a reason I'll make clear in just a second. So applying this to metric ordination, we did principal components ordination. Essentially, this data set groups into 3 different groups, loosely associated with 3 different virus genes. NS3 and NS5B are associated with hepatitis B. At the top, WSHBV is the summary of all 4 hepatitis B probes, which were essentially all collinear. And this data is largely driven by vitellogenin. We did subsequent downstream analysis and found that it actually contributed 71% of all of the dissimilarity in this whole data set, meaning it was skewing everything dramatically towards vitellogenin or the lack of vitellogenin, and we needed to do better to clarify that relationship. That is a statistical outlier and therefore had to be removed from consideration. Upon removal of vitellogenin, things clarified a little bit more. We were able to kind of get a little bit more discrimination between fish that had very high copy numbers of white sucker hepatitis B, as indicated by the points over on the right. But also, interestingly, expression of NS3 and NS5B, both genes within active hepatitis C infections, were not ortholinear. So they were actually orthogonal to each other, meaning they were expressed differently and they had different distance metrics. So something interesting going on there with the expression of those 2 respective genes. But in general, this gives us some inference to be able to say, hey, we've got co-expression, at least similarly linked expression between these 3 different trends, either associated with NS3, NS5, or the white sucker hepatitis B gene suite. And notably, this was true. A couple of different things that happened, which ultimately means I should probably rephrase my talk, is that male fish with co-infection of both Hep B and C had a lot of different genes that were differentially expressed compared to the others, and all significantly higher expression. These were either genes associated with precancer or tumor formation or general antiviral response functions, suggesting that potentially this co-infection could increase a heightened immune response. So perhaps the title of the talk really should have been that sex and hepatitis viral infection state influences gene expression and will be taken into consideration. Also notable that NS5 didn't really have any significant patterns that popped out with subsequent analysis of covariants. So NS3 related genes typically had more obvious signatures, at least as far as statistical power. Unfortunately, this is just the tip of the iceberg, and a lot that we're still unpacking as we work through this data set. So there's a non-zero chance that any questions might have already been answered, and just short on time and short on mental bandwidth to be able to convey that information. But in summary, we were able to use both qPCR and nanostring analyses to detect hepatitis B and the novel Hepasi virus and white suckers. Male fish in general had altered immune gene expression, but it was more pronounced within superinfection of Hep B and C. There's some bizarre external pathology correlation that exists, but that link is currently unclear, as at least demonstrated by the external pathology data and high copy numbers of hepatitis B. But we did get some co-infections. We are dealing with some fish that had both Hep B and C, so we have yet another model organism on our hand to be able to study simultaneous infection of hepatinovirus and hepaciviruses. So hopefully moving forward, this suggests a model organism and hopefully opens the door to continue to refine our sampling methodology and allow non-lethal detections of both of these novel viruses and general biology comparison to see what we have going on. Just wanted to give a few thanks to folks that would not be possible to have conducted this work. And I don't know if I have any time for questions. I know I was kind of burning through time there at the end.
Excellent job, Clay. Thanks for the very interesting talk. I'm so glad to hear you guys are still working to um, understand that emerging group of viruses. Folks, we got time for a quick question if somebody has one. You can drop it in the chat or unmute yourself. Okay, let's continue on. Thanks a lot, Clay.